from sunlight. So we're looking at sunlight and how plants are using it in addition to, we've already talked about photosynthesis, but now we need to think about how else do the plants interact with sunlight and what can they do with it in order to adapt to their particular habitat. And so we're going to be thinking about photoreceptors in this context. So when we look at sunlight, the idea of the sun's energy striking the plant body, and we've been focusing a lot on leaves for the idea of photosynthesis, and we're going to stick pretty much with that for the most part, thinking about, okay, what else are the plants doing when they are having the light from the sun striking their body, or we can think about artificial light too, but most of this is phrased in the sense of sunlight energy coming in. So we need to think about photoreceptors, light receptors, and what those are. And so they're molecules that the plant has in order to detect sunlight. So this is different than what we're thinking about for photosynthesis, where we have chlorophyll pigments that are absorbing light energy, and then the chlorophyll loses an electron. That's different. That's a different idea. With photoreceptors, we're going to see it's a different mechanism in how they respond to the sun's energy. So instead of an electron being given off, like what we see in chlorophyll, that kicks off that electron transport chain, with the photoreceptor idea, you tend to see an actual physical conformational shift in the molecule. And then that is a mechanism that kicks in usually some kind of a signal cascade into the cell, or ultimately off into the DNA, to allow for some kind of response. And so they call that response a photoresponse to the light, okay? Light response. So it's a fairly simple mechanism when you come down to it. It's this photoreceptor absorbing light energy, so a photon of light energy. Remember, sunlight energy acts as photon type, particle type interaction when the sun strikes a physical substance like a leaf or a photoreceptor. And so it's photons that we're thinking about of light energy. So they'll absorb that and they use that as a signal. These photoreceptors typically consist of a protein, and then there's usually some other non-protein molecule that's attached to it. And they often call that the chromophore. Is the non-protein part. They'll say the chromophore. You don't necessarily need to know that word, but just I'm more concerned that you understand it's a protein, and then there's a non-protein molecule attached to it. So really what we're talking about is the ability of the photoreceptor to shapeshift. It's able to morph or change its shape, and that action kicks off usually some kind of signal cascade so that the plant can interpret that information and act appropriately. So one of the classic examples that's given to demonstrate how sunlight energy can drastically influence the plant's behavior and response is this idea of what do you do when you put some seedlings in the light versus what if you keep them in the dark. And so that's a classic experiment. The results are being shown here in this image, where you have the plants on the left were exposed to light, and they are showing the normal growth pattern. So the fancy word for that is photomorphogenesis. So photo, light, morpho, shifting their shape, or I guess not really shifting their shape. I should say it's referring to the shape. Genesis is to create. So photomorphogenesis, what they're trying to say by that term is that these plants are displaying photomorphogenesis. They responded to the presence of light by growing up in height a little bit, but you can see they've really greened up. Their chlorophyll is fully active. If you compare it to the batch on the right that were in the dark the whole time, you can see a big difference where they are experiencing a response that's called etiolation. Etiolation. And that's the term given for especially what tends to happen is this really long stem growth. They shoot up in height because in essence what the plant is thinking, so to speak, is if you put them in a dark cupboard, they don't know they're in a dark cupboard. They think I'm getting shaded out by other plants around me. So if I want access to sunlight, I have to grow tall. And so they keep growing up and up and up, but unfortunately they don't get that they're stuck in a cupboard, and that's not really going to work. And so you tend to see this very classic definite increase in height compared to the ones that are in light. And then they don't green up because they're not getting hit with light energy to get their photosynthetic machinery up and running, their chlorophyll fully established. And so that's a very classic response. The good news is if you do this experiment, whether it's on purpose or accidentally, where you forget some plants in a cupboard, if you put them under light or out in the sun, they will green up and they'll survive. But the problem is, depending on how fast they grew their stem can be very weak, and it can take quite a while for them to strengthen that up. So don't put them anywhere windy because they'll probably flop over and stems might break. But if you see etiolated plants, it's not death to them necessarily. You can rescue them if you get to them in time. You just got to kind of baby them a little bit. Okay, so that's classic. Here's another classic response that you see a plant exhibiting where they're moving in the direction of unidirectional light. So from this image, as you're looking at it, light is being shined on them from the right side of this image. And this is a seedling of corn, Zea mays. And so this is the coleoptile is just the initial covering of the seedling as it's coming out of the soil. It has a coleoptile, so it's just referring to a baby corn seedling. So what's happening here is initially when it comes out of the soil, it's straight, but then it starts to sense that there is unidirectional light coming from the right. And so it's going to start to bend. So this is a time lapse showing that gradual bending toward the light. So you can do this with just like bright white light. You can do this. And so the scientists were wondering, like, we know white light, it breaks into a prism. You know, when you shine it through a glass prism, it breaks into all the different colors of the rainbow. So they were wondering, you know, just like sunlight energy coming from Earth, you have all the different qualities. So they wanted to know, is it a specific quality that the plant is responding to, a specific wavelength? And so they just did a very simple experiment where they did break that white light into the different colors. And, you know, you did a similar thing in lab where you were putting the glass colored lights in front of the projector. And so basically that's what they were doing with different coleoptile baby corn seedlings. And they would just shine red light, just shine orange light, just shine yellow light, and so on and so on and so on. And then they wait to see what photo response they would get. And you can see with this experiment, they really didn't get much of anything until blue light, that quality of light. So that's why you see here, I have blue quality. They know that it's actually the blue quality of light that plants are most sensitive to. And in this case, it causes what we call phototropism. I do need you to know that term. If you've had Plant Kingdom, that's something you've learned already. Tropism is the idea of movement, usually toward or away from a stimulus. It just depends. And then light, of course, they're responding and moving toward light in this idea. Here you can see on the top, you can see that relative effectiveness of light. You see it really spikes in the blue quality of light. So that nanometer, you know, 450-ish nanometers is the wavelength that plant is responding to. We'll talk more about that for light energy. absorption, it's really the blue quality. So here, phototropism, you can see, I think, if you just compare these two figures, that they kind of lay up on each other really nicely. They line up with each other. And so here what we're seeing is the absorption spectrum and the action spectrum. So if it matches, what they're saying is the pigment, here it's phototropin photoreceptor. When you expose that to light, you get this type of absorption spectrum, and then the action spectrum of getting the seedling to bend, you see it matches. So what that's simply saying is that it's due to the phototropin photoreceptor absorbing the blue quality of light. It's causing, in this case, the seedling to bend toward the light. And so it's just a very simple experiment to show that it does match to this particular pigment. So they say it's the phototropin that's really doing the photoreceptor work and getting the plant to respond appropriately, which is to bend toward the light, which if you're a plant, kind of makes sense. Remember, plants like to have their leaves 90 degrees to incoming sun rays. And so if the sun is only coming from the right, they would want to put their stem or their leaves to 90 degrees of the incoming light. So if you've driven by, the classic idea of sunflowers in a field, how they face the east and wait for the sun to rise, and then they track the sun all night long or all day long until sunset. So again, thinking about sunlight, we need to remember, of course, that you have the different qualities, different wavelengths of sunlight energy that are striking the plant body. And they, depending on the photoreceptor, will pay much better attention to certain qualities compared to others. Phototropin, we just saw, prefers blue. So another simple experiment you can do to look at how sunlight can impact the plant's response and behavior. This is an example that's super critical to plants, which is whether or not the seeds decide to germinate. And so again, this is with lettuce. Lettuce is famous. If you've ever bought lettuce seed packets, it usually says on the seed packet, do not bury the seeds, just put them on the surface of the soil, because they actually need exposure to sunlight in order to germinate. And so this is a very simple experiment. They have a Petri plate with a wetted filter paper as background, and then they place the seeds on that, the lettuce seeds, and then they would either keep them in the dark, like those, or this middle picture, what they did was they exposed the plant or the seeds to red light. And you can see, so red light is part of sunlight. Almost all the seeds germinate. But this is interesting. Then they said, okay, let's take another set of seeds. Let's expose them to red light for a set amount of time and then switch and expose them to far red light right after that. And when you do that, you see most of them don't germinate. Then they said, well, let's play with that again. Let's expose them to red light. Let's expose them to far red, but then let's go back and expose them to red light. And when you do that, you see the seeds germinate. So this was an interesting experiment. And initially when they did this, they're like, ooh, this is weird. What's going on here? And so you'll often see it explained as the plants only respond to the last wavelength they're exposed to, or they only remember the last thing they were exposed to. And so we know that red light coming from the sun stimulates them to germinate. Far red, on the other hand, actually has the opposite effect. It keeps them from germinating. But if you expose them one more time to red light, that's what they remember or will respond to, and they'll go ahead and germinate. So this experiment really got them thinking about what is going on here in terms of it. It seems like they can toggle back and forth in what's going on here. And they started investigating what kind of photoreceptors might be allowing for this to happen. And that's when they stumbled on phytochrome pigments. And so again, if you've had Plant Kingdom, this should sound vaguely familiar. And with phytochrome pigments, again, we're looking at this idea that it's a shape-shifting molecule that it will, shift its shape in response to different qualities of sun. So photoreversible, it's very interesting. So there's alternate forms of this pigment showing up in the plant body. And it just depends on if that part of the plant body got hit with red light or is it getting more far red light. And actually what this pigment is doing, so there's lots of phytochrome pigments throughout the plant body, primarily in the leaf tissue, they think, or in the leaf buds in the spring. And really what they're doing is they're assessing what version of my pigment am I experiencing. And so there's alternate forms of the pigment, and they actually kind of assess the ratio. So let's work through this. So there's a so-called P sub R form of this pigment. So that's called, the R stands for red light. So phytochrome red, PR, phytochrome red. So this will be present when, okay, let's say the plant has P sub R in its leaves. And then that gets hit with red light. What happens is it causes the P sub R form to shift into the P sub FR form, which stands for far red. If there's P sub FR form in the plant body and it gets hit with far red light, it converts it back to the P sub R. So what the plant is doing is it's saying and assessing how much P sub R version of phytochrome do I have in me? How much P sub FR do I have in me? Excuse me. So it assesses the ratio of those two forms of the phytochrome pigment. So this P sub R and P sub FR, it's just referring to the different forms of this phytochrome pigment. And so the ratio of these two will signal to the plant to kick into a certain response based on that ratio. So you could say that it leads to a shift in phenotype. Often it's referred to as a response or a behavior that the plant will exhibit. So this is actually pretty simple mechanism. It's just taking this pigment and having two alternate forms of it. And that ratio of those two can really tell the plant quite a bit of really crucial information. And being, am I in the sun? Or if a plant is exposed to a lot of red light, by the way, if they are absorbing a lot of red light, that's telling them, ooh, I have really good access to sun. I have a lot of sun beaming down on me right now. However, if they're experiencing a lot of far red light tends to percolate down, like if you have a forested ecosystem. What tends to happen is in the lower canopy, under the upper canopy, anything down on the forest floor or midway in the canopy system, they get more far red light. Because what's going on is, if you think about a complex forest ecosystem, the plants that are tallest, they want red light for photosynthesis. So they're snagging that, they're absorbing that red light. That means if they absorb a bunch of red light, less red light comes down because they've absorbed most of it. So they kind of hog the red light up top and they don't care quite as much about far red. So they'll just say, yeah, that can go through. I don't need to absorb it. And so more far red comes through. So in a complex forested ecosystem, this ratio of red to far red can be really crucial to all the plants in that ecosystem. It's really giving them important information. So if you're a plant in the mid to low level canopy system area, that would say, okay, I'm experiencing far red light. I'm probably getting shaded by some nearby trees that are getting taller than me. And depending on who you are, what kind of plant you are, that could be no big deal. If you're a shade adapted plant, being hit by far red light's fine. You're shade adapted, so it's not a big deal. But if you are a sun loving plant and you're getting the message, I'm getting a lot of far red light, I need light to stay alive because I'm a high I'm not shade tolerant. I need a lot of light. And so your behavior, if you were a plant where like, oh my gosh, I need a lot of light, but I'm getting far red light hitting me, that means I'm being shaded. So your response would probably be to grow tall. You're trying to get above the canopy that's shading you out. So it just depends if you're shade tolerant or not, how you're going to respond. But really what you're doing is you're assessing the ratio of the phytochrome. So if you are in the lower canopy, and if you're getting hit with a lot of far red light, what's happening is that you're going to actually get a buildup of PSABR in your body. Because if you keep getting hit with far red, that means your PSABR is going to get converted to PSABR. And so you'll say, oh, I have a lot of PSABR compared to PSABFR because I'm getting hit with so much, far red light. So my PSABR is high, my PSABFR is lower. That means I'm being shaded. And so this ratio is very important. Here's a visual. I think this might make it a little easier to think about this kind of shifting or toggling back and forth. So the phytochrome pigment that's in the so-called red form, that means they call it the PSABR because they know that it will preferentially absorb red light when it's in that form. And so it'll absorb the red light and then that converts it into the PSABFR form. Here they've written active form. What they're often referring to here is flowering. They often talk about flowering as kind of the biological response to the active form. I'm more concerned that you understand, think about this idea where if the phytochrome red version of the pigment absorbs red light, it converts it, changes shape to the PSABFR form. And then if that starts to absorb far red light, it toggles back or converts back to the PSABR. So it's how much of the PSABR versus the PSABFR version of the phytochrome do they have? And that will tell them what kind of biological response they need to do. And so a lot of the work that's been going on in this field of research is geared toward understanding when a plant decides to flower. Because of course it's very important for us because so much of what we eat comes from flowers that get pollinated and turned into fruit, right? And so they've been very, very focused on how do plants decide when to flower. And if we better understand that, we can better grow plants for food production. So here's another way to think about it, just trying to get you to think about the PSABR and the PSABFR. They're just different versions or shapes of the phytochrome pigment. That's really all we're talking about. All right, so here they've drawn the PSABR form of the pigment. And there's the non-protein part, that's the so-called chromophore. And then you can see when the PSABR absorbs red light, it changes its shape. The phytochrome pigment just changes its shape. And so very often what they see is that the PSABFR form often is responsible for a signal cascade that ultimately affects gene expression. And so that's what they're trying to show here in this drawing, that you have this signal cascade. Eventually the DNA gets the signal and you get some kind of shift in gene expression. But it's just changing its form or its shape in response to red light or far red. You can see if a piece of FR form gets hit with far red, it converts it back. So the plant is continually assessing the ratio of these two versions of the phytochrome pigment. And from that, they can get information on how much sun they're getting, what quality of sun they're getting, if they're getting shaded out. So here's this idea again of a forest canopy, or a mature forest with a canopy, multi-canopy system in place. And so the trees that are tallest, they get a lot of red light. And so they absorb a lot of that, use it for photosynthesis, the red light trickles through into the understory, the far red will come down more so. And so that's definitely going to shift what's happening. You know, the trees that are tallest will have a different phytochrome ratio compared to the trees that are in the understory. Right? So the trees up top, they're going to have a lot of PCFR inclusion of the phytochrome. But the trees in the understory, they're going to have the exact opposite. They'll have more PCFR. And so it's really, when you think about it, pretty straightforward mechanism. They're just looking at having the two different shapes of the phytochrome pigment, and they just assess the ratio. But it can give them such crucial information. Here, this is just showing the different, so the blue is all the sunlight energy. When you think about, you know, the sun striking the earth, so to speak. The red line, let's see, phytochrome, absorption spectra of OAT phytochrome is the red, PCFR is the red line, PCFR is the green line. So you can see they're absorbing in the red and the far red, just to get you thinking about the different qualities of light that they're absorbing. And the blue line is at the very, so when the sun is striking the earth or the very top of an ecosystem canopy, that's what they're getting hit with. And again, you know, they're going to preferentially absorb the red. And then they talk in the notes about the far red coming through the lower canopy. Right? Thus, the relative proportions of P sub R to PFR are determined by the degree of shading in the canopy. So in a mature ecosystem, you definitely have plants experiencing different ratios of phytochrome pigments for sure. So let me look at the, okay, let's go to this one. So here we're looking at, so the main focus initially for phytochrome was flowering. When, you know, what's going to cause certain plants to flower. And in Plant Kingdom, we talk about the idea that there's some plants that are so-called long day plants, short day plants, day neutral plants. And so it's just looking at, again, you know, if they're long day plants, they need some day length longer than some critical period in order to trigger into flowering. Short day plants is just the opposite. And so with this idea, you know, they were very intently looking at flower, you know, how does a plant decide to flower initially? Now they've branched out into other, you know, what else is phytochrome covering? What else is it responsible for? And so I just wanted to put this here to show you that they're fleshing this idea out some more. It's not just flowers. They're looking at just general growth, production. We'll talk a little bit about plastic, a little bit, germination of the seed. Here you're actually seeing in P that it inhibits internode. What they mean is it just makes the stem shorter. If it's inhibiting internode elongation, I don't know why they have to say that, but it's just saying that the stem stays shorter. That's the upshot. It's not as long. De-etiolation just means you're converting back out of that very sad yellowing plant with tall stems. Okay, so they're branching out into different effects that phytochrome has. One of the students that went to Northern, Jeremiah Wagner, he was a botany major here at Northern, and he was part of the research team that published in the journal, I think it was Science or Nature, one of the big wigs. One of those, both of those journals are huge. If you get a publication in one of those, that's a huge deal. And so he was one of the, he was a PhD student at the time, and he helped break open the knowledge on the physical structure of phytochrome, the actual pigment. And so it's kind of fun to think how initially when he came to Northern, he grew up on a farm, and I was his advisor. He's like, I don't want to do anything with plants. I'm so sick of plants. I had to farm, you know, I was slave labor for my parents my whole life. But what did he do? He ended up switching to botany and got his degree, and then his PhD on phytochromes. I had to laugh. So never say never. Okay. So another idea about photoreceptors, another type, are the so-called cryptochromes. And they've been doing quite a bit of work at this too. This is a blue light. So we shifted out of red and far red. We're looking at blue light again with cryptochromes. And so there's a few ideas here that I'll get into. So cryptochrome, if they absorb light, one of the responses that they can do is to stop growing in stem length. So it suppresses stem elongation, just means that they aren't being stimulated to elongate their stem in any way. It slows that down. Circadian rhythms, which is just thinking about the idea that all organisms seem to have this day-night rhythm that we respond to, and so they think that the cryptochromes are essential in helping the plant understand that circadian rhythm, so to speak, and respond appropriately. Pigment production, so anthocyanins are pigments that you've already heard about, and we'll talk more about them actually through the semester. So it can stimulate anthocyanin production when the cryptochromes absorb blue light. It can tell the plant to start making anthocyanins. And now they're finding that photoreceptors often complement each other, where you can have the phytochromes and the cryptochromes kind of sharing signaling in order to accomplish some kind of phenotype or shift in gene expression. So the bottom image is, again, just showing this shape-shifting idea that when you have the so-called inactive form of the cryptochromes, they have one shape. And then when they absorb blue light, they change their shape. And then again, you're showing how once that shape has changed, it can cause a cascade response or signal until you get the ultimate response, which is usually some kind of shift in gene expression. So that's what they're trying to show on the right side of the figure here. Oh, solar tracking. So when you do drive by sunflowers in the field and you see them tracking the sun, you can bet that they have a good dose of cryptochromes that are involved in that process. In the notes section, I just wanted to make you aware too, cryptochromes, it says they're found in many organisms, not just plants. Algae, cyanobacteria, they found them in fruit flies, mice, and humans. So we have cryptochromes too. Okay. Here again, just looking at showing stem reduction or slowing down in the growth. So here they're shining blue light and they show a slowdown in the growth rate. And so this was cucumber seedlings. And so they're just showing how you have that definite slowdown when they're being hit with blue light. Another group of blue light photoreceptors are the phototropins, which we already mentioned with the idea of phototropism. But I wanted to mention a few other things. So the initial idea of phototropism was something that was first discovered a long time ago. It was actually Charles Darwin of, the theory of evolution and natural selection. It was him and his son that got into this work. And so phototropism has been around for a long time. But they now know that it's due to the phototropin pigments that are acting as photoreceptors. But now they've, of course, looked at, well, what else can it do? Guard cell opening is, phototropins are directly involved in that process, and that's huge. And so in the morning, when the plant is getting hit with sunlight, it's really being impacted by the blue light. So what's happening is the blue light is being absorbed by the phototropism, and it actually causes a situation that allows potassium to flood in to the guard cells. There's a little bit of ATP necessary to get this to happen, so it's a little bit of a cost, but because that phototropin pigment is absorbing that light energy, it sets off that signal cascade. Okay, spend a little ATP, get potassium flooding in so we can open our stomata. All right, and so it's a boom, boom, boom. It's very quick and very effective. Phototropins also help the leaves orient to the sun as well. And that makes a lot of sense that they have some easy mechanism in place that they can use to get the stem, the petiole of the leaf, to rotate and shift so that the leaf blades like to be 90 degrees to incoming sunlight. Chloroplast movement, I'll show you a couple pictures of that. So within each cell of a leaf, there's usually lots of chloroplasts. And depending on what's going on in terms of light intensity, we see that chloroplasts will move around in the cell to optimize access to sun, or if they're getting too much sun, you actually see chloroplasts lining up in single file to hide from the sun. It's that level of sophistication where the chloroplasts in each cell can physically move to get better access to sun or to hide away from too much sun. It's pretty amazing. Seedling phototropism, which we've already talked about, that corn coleoptile experiment, as I talked about, that was, Charles Darwin and his group did that way back in the 1800s. So he didn't know exactly how it happened, and we will talk about the mechanics of that when we get into hormones, which will be our next topic. But it's actually the auxin hormone in the tip of the coleoptile is actually, with that unidirectional light, it's actually stimulating the auxin to shift and just flow down one side of the stem, which happens to be the unlit side or the dark side. And so Charles Darwin didn't figure that out. It took a long time for that to be figured out, but we'll talk a little bit more about that in the next section on hormones. But again, just the visual, trying to show you that it's a shape-shifting idea. So it's a pretty simple mechanism. In their before they get absorbed or hit with blue light, they look one way physically, and then that absorption causes that shift. And then that usually leads to a signal cascade that they're showing going off to the right. By the way, protein phosphorylation, we'll talk a little bit more about this in the next section. That is a way to send a signal. It's a pretty simple mechanism. They just transfer phosphate groups to donors and acceptors, essentially, kind of like electrons getting transferred, except it's a phosphate group. And that's a signal that will tell whoever gets that phosphate to go ahead and do something. So here, again, just a visual trying to show this idea of unidirectional light, and it's actually the blue light quality that will cause that phototropism. So auxin is being produced in large portions in the apical meristem. And what's happening is it's flowing down the dark side, and it's causing those cells on the dark side to grow. So the dark side of the stem is grow. The side that's getting hit with blue light actually slows down its growth, and that's what's causing that bending. And so it's an asymmetrical growth response in order to get the leaves to be 90 degrees to the sunlight, or the blue light in this case coming in. Here's an idea with the chloroplast movement that the phototropins can help with. So when you look at chloroplasts in a single cell and you start exposing them to really shady conditions, they will kind of all line up in a way that maximizes any light they get. They can try to capture it. And so they physically orient themselves to try to get as much quality, as much absorption that they can. Highlight, on the other hand, they kind of line up single file top to bottom so that they can hide from too much sunlight. Because if you get too much sun, we already learned that can start to damage, create free radicals, cause damage. All right, so they actually hide away. I mean, that's pretty amazing that they can do something like that in order to deal with these darkness. They tend to see, they just kind of sink, almost like they're sleeping, like, I guess we'll just hang out. But yeah, pretty amazing. Here they're showing again the linking the absorption of phototropin pigment to the movement of the guard cells. And again, they're just showing where you see those peaks in the blue light quality. So again, phototropin pigments absorb blue light and we give that kind of absorption spectrum. So it matches up really well with stomatal opening. So now stomatal opening, just so you know, it's complicated. I'm not going to get into the details, but I did put this up just to impress on you that I'm giving you the light version of what's happening when stomata open. All right, there's a little bit more to it, but you do see the blue light coming in here in the phototropin pigment. So that's a good visual for you to think about. I want you to know that. All right. And then we've already talked about back when we were looking at stomata and guard cells that it's actually potassium that's getting pumped in early in the day. So hang on to that thought. You've already learned that. So here they're showing the potassium coming in. All right. And then eventually, midday, you start to see sucrose taking over that role to make sure that the stomata stay open. But again, it's just playing with water potential. If you dump a bunch of potassium or sucrose into the guard cells, that's going to cause water to rush in based on water potential. And when you have water rushing into those guard cells, then they move away from each other and reveal the cord. But what's kicking that all off is blue light. So those phototropins are capturing that blue light, absorbing it, and that sets off. So what they're trying to show here with the artist is that you get this signal cascade now from that absorption of blue light by the phototropin, and that leads to this step-by-step signal cascade until ultimately you get in the morning a flood of potassium coming in. Okay, that's pretty much all I want you to know on that. You don't have to get into all of the details here. And this is just looking at the co-action. So again, getting you to think about the idea that now they're realizing these different photoreceptors can interact with each other. And in that respect, they can send off pretty sophisticated signal cascades to help the plant fine-tune their response. So it's a very appropriate response to the data they're taking in, so to speak. Okay. So that's really all I want you to get from this, is that there's interactions between these photoreceptors that can lead to very sophisticated signal cascades. Here, UV light for us or any organism, not such a great thing in high doses, as we know. Plants, though, they can use this information. Ultraviolet light is actually a source of information for them that can be important because they do have certain receptors that will absorb ultraviolet, and it can give them important information. So for any living organism, UVB is the wavelength that's particularly damaging to cells and tissues. Plants actually have photoreceptors for UVB, and it's called UVR8. And it's a funky dimer protein that has some weird salt bridges. And what's happening is when it absorbs when the amino acid here, the tryptophan, when it is sensing that UVB light, what happens is you get those salt bridges, they break. So this is a little weird. And then what happens is that the dimer breaks into two physical monomers. So this is a little different than all the other photoreceptors. It's a little bit more drastic in the sense of it's just not changing its shape. They're actually dimers that separate physically. So that's an interesting find. We're not expecting that, but anyways, it works. And again, it leads to a signal cascade that will alter gene expression. Very often what it will do is cause the plant to shift away from that ultraviolet light. So they will try to hide away. Some leaves will actually dip down if they're getting exposed to too much sunlight. They'll actually try to have their leaf blades. So if it's high noon, most leaves are horizontal to the ground so that their leaf blades are 90 degrees. But if you're getting hit with a lot of UVB light or ultraviolet light in general, sometimes what you'll do is you'll orient your leaf so it's parallel to the sun rays. So you could either move your blade up, or you might move your blade down, and that can allow for the sun rays to flow past you, and you'll get less exposure to the ultraviolet light. So too much sun, which includes ultraviolet light in that sunlight, can be a problem for plants too. Okay, so this is a very short slide set. This is like a brief introduction to photoreceptors to get you thinking about, we focused a lot on photosynthesis, which is important, certainly, but plants do other things with sunlight that you need to be aware of. And so I was trying to give you a taste of that in this slide set, in this information. And then some of this will kind of come into the next slide set. We're going to get into kind of a little bit, kind of a general introduction to signal cascades. So this idea that's being shown here on the right, what is that all about? So I'll give you the basics of that. We don't have time to get into in depth. This is not a molecular biology course either. So we'll get into the basics, and then we'll look at hormones and how hormones are very heavily involved in getting these signal cascades run to completion. So that's the next topic that will start on Friday. But we'll stop here today.